Hi there. Welcome to the first episode of Sci Herald, a YouTube channel that briefly presents some of the latest published science advances. Our goal will be to present some scientific publications picked up from the past 1 or 2 months. The links to presented papers will be available in the video description, together with any possible resources to help understand them. So let's go ahead and see some of the recent science stuff. Our opening question is, could brain use light to communicate? That's a question answered in this paper published in the journal Scientific Reports. Researchers in Canada theoretically discussed the possibility of neurons transmitting light signals between one another to communicate between cells. This study evaluates how light waves ranging in between infrared and orange could actually be transmitted through neurons, much like an optical fiber. They computed how such light waves could propagate in the myelin sheath, discovered around neurons' arms called axons. Taking into account various imperfections, researchers concluded that it is actually possible that light signals are used for brain communication, and proposed methods to verify the predictions in experiments. Now, it is up to the experimentalists to check whether this happens or not. Another recent multidisciplinary publication, also on Scientific Reports, targets cancer cells. Korean scientists used terahertz frequency electromagnetic waves to analyze DNA of cancer cells and found cancer DNA fingerprints that may be used for fast, precise, and early identification of cancer cells. The difference between normal DNA and cancer DNA explored in this work resides in the epigenome, in this case DNA methylation. This picture shows the schematic difference between healthy DNA and cancer DNA, where the red tags attached to some spots are methyl groups linked to them. These methyl tags may have various functions, but cancer DNA has way more tags in specific places, as shown here. Usually, complicated chemical reactions are needed to identify whether these tags are there or not, but the new method may allow one to just scan the DNA with terahertz waves and know whether it is a normal DNA or a cancer DNA. And this can possibly be done much faster and cheaper than the standard methods, and could become a big deal in cancer diagnosis in the future. And not only cancer. For example, type 1 diabetes was recently linked to increased amount of such methyl tags on this study that recently appeared in Nature Communications, and perhaps could also benefit from this kind of terahertz techniques. Talking about DNA and cancer, a related piece of news is attracting attention. Researchers in China used CRISPR-based gene editing in human treatment for the first time. For this, they knocked out a gene from the DNA in some of the patient's immune cells to make them able to fight cancer cells, using the now famous CRISPR-Cas9 technique. If you don't know what CRISPR-Cas9 is, it is a revolutionary gene editing tool discovered and developed in the last 20 years or so. In short, a protein called Cas9 is used to cut DNA very precisely and cheaply, and then a desired DNA string may be attached where the DNA was cut, though in this case a gene was just targeted with no replacement or addition of new gene. If you want to know more about CRISPR, I recommend Kurzgesagt and SciShow videos. Their links are in this video's description. But way before humans, nature has done genetic engineering for billions of years with evolution, and we are still trying to understand some parts of this process. For instance, how did animals get bones? Researchers may have found some evidences of what happened by studying rocks in Siberia. In this paper published in the journal Geology, scientists report that over 545 million years ago, sea sediments held large concentrations of magnesium compared to calcium, with little oxygen present, forming a mineral called dolomite abundant. But around 545 million years ago, more aragonite and high magnesium calcite appears, signaling larger amounts of calcium and oxygen, in the same period as the first skeleton fossils appeared. The amount of calcium kept growing, and around 520 million years ago, low magnesium calcite appeared, together with low magnesium calcium skeletons. The researchers suppose that as oxygen levels rose and weather conditions changed, calcium was washed away from continents to the ocean, and the higher calcium to magnesium ratios favored the biological development of calcium skeletons between 545 and 520 million years ago. Finally, we go from the sea to space to talk about black holes, one of the most mysterious objects in the universe. Earlier this year, we had the detection of gravitational waves coming from two black holes colliding, but besides that, Hawking and collaborators published one interesting paper on hairy black holes. They attributed soft quantum hairs to black holes as a way to account for what happens with information that falls into a black hole. This so-called soft hair is actually composed by light or gravity particles, known as photons and gravitons, of near-zero energy close to the black hole's surface or horizon. And since nothing can escape a black hole once it falls in, it seems that information is lost forever when it falls into one. But physically, information can only be transferred or created, not destructed. This soft hair could be where information is trapped. Now, another study shows that one can shave these black holes and care not about its hairs as long as it is properly dressed instead. More precisely, common states of finite energy can be dressed in a way that the soft hair doesn't really matter. But shaving the black hole actually restores the information paradox, suggesting that a fluffy black hole is not enough to understand what is going on yet. So the mystery still remains. Thank you for watching, and if you are interested in future videos like this, subscribe to this channel. And since this is my first video, comments are welcome to improve the channel and its production. Hope to see you soon. Bye!